So, hello everyone. The work that I've been doing in in the past 3 years now followed from a bachelor's in civil and environmental engineering, and which I pursued at Cardiff University, and then I liked Wales so much that I decided to stay for another 3 or more years. Um, so I've been looking at developing self-healing mechanisms in geotechnical structures. The project that I've been working on is part of the Materials for Life project, which focuses on mainly on cementitious materials and concrete. And the Materials for Life project is a collaboration between Cardiff University, Bath, and Cambridge. What our vision is to draw inspiration from nature and the natural human body mechanisms for self-healing, and apply those mechanisms to construction materials. So the work that I've been doing is aimed at geo-environmental applications. So why do we actually need soils that can self-heal themselves? For a few reasons. Because of growing population and urbanization, our infrastructure is becoming ever more complex, is becoming bigger, and many of our infrastructure projects are largely earth structures. These can be of a huge scale, and damage repair to those structures can be extremely difficult and expensive as well. It has been estimated that around 20 million pounds are spent a year in the highway sector in the UK to repair slope instabilities and embankment instabilities. So we believe that biological processes and bacteria naturally living in the soil can provide a solution to these problems. Some background to where the idea came from and information. It has long been known, I think for about 100 years now, that bacteria are actively involved in precipitating minerals in the environment. These two pictures, scanning electron microscope pictures that I have here, are of two different strains of bacteria producing calcium carbonate. And not only bacteria can precipitate minerals on the micro scale, shown in these two pictures, but they can actually do so on a very big scale. So examples are the limestone deposits at Yellowstone National Park. In fact, some geomicrobiologists postulated that all limestones on Earth should be considered of biological origin. That's how big the impact of microbiology is on on Earth geology. So some of the research that used these principles aimed at developing self-healing concrete, and the picture on your left shows bacterial concrete. So bacteria was embedded in the mix of concrete, and once cracked, nutrients were supplied to the bacteria, which was able to seal the crack completely, as we can see from the picture. Another impressive research was done in the Netherlands. They managed to cement a big mass of soil, around 100 cubic meters of sand was treated with bacteria, and that's the result that we see. So our idea was to combine these two concepts and create soils or geotechnical structures that can heal themselves. Examples of where this idea might be used is in earth structures such as earth dams, water retaining structures, or embankments. These can undergo damages in the form of cracks, and if not treated, these cracks can lead to catastrophic failure. So what we think can be done is use the bacteria naturally living in the soil, provide some nutrients to them by pumping these nutrients into the soil, and these bacteria will be able to produce this biogenic cement. So once a damage occurs in the form of a crack, and oxygen and water are available to the bacteria will wake up and close those cracks. So that's the concept of my PhD, and I started off by searching for suitable bacteria. The bacteria had to satisfy a few different criteria. One of them was to form spores. Spores are simply put a mechanism for bacteria to survive unfriendly conditions in the environment. So I'm not sure if you can see it very well. This is a microscopic picture on the left showing spores, which are the swollen bit at the edge of the bacteria that's blue-greenish. And the other criteria was for the bacteria to, of course, produce biogenic cement. And the one that satisfied all those requirements is called Sporosarcina urea. I spent a long time looking for a bacteria that behave the way I wanted it. I actually used another 3 strains. So this was the only one that natural to soil that actually worked quite well. So the first experiment I did was showing the cycle of the bacteria being able to produce the cement and then respond to damage and produce more biogenic cement. In laboratory conditions, very microbiological experiments. So I started off by culturing bacteria. I gave it the right food for it to form the biogenic calcium carbonate. In the process of the formation of this crystal, the bacteria becomes encapsulated within it. So the crystal actually becomes, it entombs the bacteria. So the bacteria starts to starve. There is a limited nutrients, so it forms a spore. 6 months down the line, I took those same crystals and I dissolved them with some acid, which exposed the spores. I then gave them some food, and they germinated. That means they came back to being viable bacteria, and they were able to produce more calcium carbonate. So that was the basic science that I had to show before I move on to a more complicated environment that is soils. So before I moved on to using the bacteria in soils, I had to perform a small optimization study and create a computer model telling me what are the optimum conditions for this certain strain of bacteria to produce the maximum, to use up all of the chemicals available and produce maximum calcium carbonate possible. But because that's not very interesting, I haven't included it in this presentation. But just so you get an idea of how the healing works, this is my experimental setup. It's shown on the top left. These are cylinders filled with sand and bacteria. Bacteria in really high numbers, about 10 billions per milliliter of liquid. And I performed the initial treatment of the soil for about almost 40 days. So I would pump nutrients every 5 days approximately, and then the bacteria would metabolize those nutrients. And I would use pH and calcium ions measurement to show me that the bacteria is active. And these zigzaggy graphs show the activity of the bacteria. So the high pH is is an indication of bacterial metabolic activity, and the calcium ions disappearing from solution meant that calcium is actually converted to calcium carbonate. And the interesting thing here is that bacteria was actually able, in this quite a long span of time, to every single time I gave it food to actually get rid of almost all of the calcium. And that's a really high concentration of calcium. That's probably the highest that this bacteria can take. So at the end of this stage, I took the samples and I physically broke them in a compression machine. So the graph of stress strain shows how different a bacterially healed sand is to normal sand, how different they respond. The orange curve shows the response of only sand, and the normal way it fails is usually by bulging out. That's shown on the on the first picture on top of the graph, and then the blue curve shows how much more the shear strength has increased. It's approximately 5 times. And how the behavior of the sand is no longer as a cohesionless soil. It's a brittle type of failure. I've tried to show it with the arrows, but you can probably see this typical diagonal shear plane occurring. So then I took some samples from the sand, and I looked at them. I looked at what the microstructure of the sand looks like after the bacterial treatment, and these pictures show. If we look at the top microscopic image, it shows a few grains of sand, which are actually bonded by these spiky crystals formed. So what the bacterial calcium carbonate is doing is actually providing cohesion and bonding between the sand particles, and providing cohesion to a effectively cohesionless soil. And the next step, I'm actually still doing this experiment. It will probably end in about 2 weeks' time. I took those same samples that I've physically broken, and I took them back to the incubator, and I started feeding them with nutrients again. So the good news was that the bacteria actually responded quite positively. The spores have woken up, and this is shown by the pH increase and calcium disappearing from solution. It's not as good as before compared to the initial treatment, but it's still an indication that there's something going on there. And at the end of this experiment would be to actually close the self-healing cycle, was to take these samples back to the testing machine and see whether the bacteria have improved and have healed the damage that I caused in the first place. Okay, that's my last slide, I think. The important findings are summarized in this slide. Is we've shown in laboratory conditions, in idealized conditions, that that the spores are survive for quite a long period of time, and they're able to retain their calcium carbonate formation capabilities, even after approximately 6 months of storage. The bacteria was able to cement a significant mass of soil and was able to actually increase the shear strength of the sand by 5 times. And I've shown that the self-healing cycle is possible in laboratory conditions, which suggests that implementation of this idea is actually possible would be possible with a little bit more work on an industrial scale in the future.
Okay, thank you.
Thank you.